Okay.
Thank you very much, Victor. So, well, you have 15 minutes to present your work, and the stage is yours, Victor.
Thank you. Thank you so much, Dr. Gutierrez. Okay, good morning, everybody. My name is Victor Velázquez Martínez, PhD student in Industrial Engineering under the guidance of Dr. Efrain Delgado. Today, I will be presenting part of my research, improving shelf life of sugarcane bagasse phenolic compounds by microencapsulation. So let's get started. Sugarcane is the most important plant to obtain sugar in the industry. During harvesting season, farmers cut the stems, and these malleable stems are transported to the sugar mills, where by using this milling train, the juice is extracted from the sugarcane stems, and the juice goes through the whole process to obtain raw sugar. But in this very first stage of the process, sugarcane bagasse comes out of this process. This sugarcane bagasse is stored in the jars of the sugar mills in huge amounts. So to give you an idea of how big is this amount, in table 1, we can see 3 different locations that were the sources of the material for my research. One sugar mill from Veracruz, Mexico, the other one located in St. Mary, Louisiana, and the last one is Rio Grande Valley Sugar Growers in Santa Rosa, Texas. So as you can see, the average production of sugarcane bagasse per harvesting season is over 300,000 metric tons of sugarcane bagasse. That is why this is a huge amount of sugarcane bagasse available for different projects. The most common uses of sugarcane bagasse are for wall cladding and wall panels, as a food for ruminants, cement additive, to obtain ethanol, but it's mostly burned in boilers to provide all the steam required to produce energy to operate the sugar mills. On the other hand, free radicals are unstable molecules that have an unpaired electron in its outer orbit. They can attack healthy cells in our body to obtain this electron to obtain stability. But our body also produces antioxidants that can donate this electron to stabilize free radicals without becoming a free radical itself. But the consumption of junk food, radiation, air pollution are factors that are increasing the production of free radicals, overwhelming the capacity of our body to produce the antioxidants to stabilize these free radicals. That is why we need to look for another sources of antioxidants. Here's an image of cranberries. As you know, cranberries have powerful antioxidant properties. We can use dry cranberries in order to make functional foods. But also, we have another option, that is extract these bioactive compounds to fortify foods to make functional foods, or use these bioactive compounds as a nutraceuticals. That is why the goal of this research is to use that huge source of sugarcane bagasse to extract the bioactive compounds and to make functional foods or nutraceuticals. That is why we need to first test how much phenolic compounds are left in the sugarcane bagasse, that is an industrial waste, and its antioxidant activity. Table 2 summarizes the results for TPC, the total phenolic content from each location, and the antioxidant activity measured by 3 different methods. In average, we can obtain over 5 milligrams of gallic acid equivalents per gram of dry matter. DPPH inhibition, we can obtain over 6 Trolox equivalents per gram of dry matter, over 12 Trolox equivalents per gram of ferric-reducing antioxidant power, or from 58 to 82% of inhibition of free radicals measured by SOD test. Also, by doing some calculations, the theoretical yield of phenolic compounds per sugarcane production per sugar mill is over 2 metric tons of phenolic compounds if we use the total sugarcane bagasse production. Then we test for the antimicrobial properties of these sugarcane bagasse extracts. In order to do that, we use 5 different bacteria and 4 different modified yeasts. Here are the implications. For example, for modified yeast, they are the human homologous for different diseases related to cancer, such as lung cancer, colon cancer, skin cancer, and Werner syndrome. And the bacteria for diarrhea, infections, and so on. We use the microplate technique, and we use MTT as an indicator of the microbial growth. Here, the dark blue color indicates that there was a microbial growth, and this colorless reaction indicates that the sugarcane bagasse extracts were successful inhibiting the growth of the bacteria or modified yeast. Figure 7 summarizes the results for this against bacteria. We also use 2 antibiotics for comparison purposes. We use ampicillin and kanamycin. And here we can see that the sugarcane bagasse extracts were successful against Bacillus cereus and Staphylococcus aureus, but just using the sugarcane bagasse extract from Mexico. Texas was the second one, and Louisiana showed almost no effect against any of these bacteria strains. So the minimum inhibitory concentration was found at 200 ppm for Bacillus cereus. Figure 8 summarizes the results for against modified yeast. In average, all sugarcane bagasse extracts were successful against SGS1, RAT14, and MHL1, a concentration of 400 ppm. This is interesting because it shows that it has some effect against colon cancer, skin cancer, and Werner syndrome. So that is why we can look further for future research using real carcinogenic cells. After testing the antioxidant, the total phenolic content, and the antimicrobial properties of sugarcane bagasse extracts, we look also for a technique that will help us to protect these bioactive compounds against factors such as light, oxidation, pH, temperature. That is why this is the table for shows us the experimental design used for this purpose. The independent variables for spray drying technique was the inlet temperature, the ratio between maltodextrin and codon-seed meal protein as encapsulated agents, and obviously the core material was the extracted bioactive compounds for sugarcane bagasse. The response variables for this design was the encapsulation efficiency, the total phenolic content per 200 milligrams of microcapsules, and the antioxidant activity measured by DPPH and ferritin-reducing antioxidant power. We did all the treatment combinations, and we measured all the response variables, and then we did the statistical analysis. Here, factor A means that is the inlet temperature, and factor B, the ratio of the encapsulated agents. As we can see with the, using the p-values, the inlet temperature was not significant on any of the parameters of the response variables, but the ratio of the encapsulated agents was significant for encapsulation efficiency and for FRIP. So after testing all these statistical analysis, the optimal parameters was to use 63% of maltodextrin with 36% of codon-seed protein, codon-seed meal protein, using an inlet temperature of 125 centigrades. Here in this table, we can see the low end of the prediction confidence interval and the high end of the prediction confidence interval. After testing again all the response variables using the optimal treatment, we saw that all the parameters, all the measurements were within the confidence interval, validating our model, and we can assure that this is the optimal treatment for this experimental design. After that, we test for the shelf life of phenolic compounds. First, fresh extract. Using a multi-pair test, time zero means that we measure TPC and antioxidant activity in fresh extract. Time one means that we use the same fresh extract after 1 month under storage conditions at 4 centigrades. And we can see that the hypothesis was rejected. There's no equal, but we saw a small increasing instead of decreasing. So we can assure that there's no degradation of any of these parameters under storage condition that will help us to, will give us a time window to process these fresh extracts before microencapsulation. Also, we did an accelerated thermal stability test using the microcapsules. And for comparison purposes, we use also free phenolics. Here, graph A shows the free phenolics, and in this graph, we can see the microencapsulated phenolics. We use 3 different temperatures, 60, 80, and 100 centigrades. Clearly, we can see degradation using 60 and 100 centigrades, and we can see an increase in this. We can answer this after, or just to finish the presentation. But clearly, free phenolics show degradation, while the microencapsulated phenolics show stability. So in this manner, we can see that the microencapsulation helps to protect the phenolic compounds against temperature over time. We did also the characterization of the microcapsule by scanning electron microscopy. Image A and B are for the optimal treatment, and for comparison purposes, we also use one of the non-optimal treatments. Here for optimal treatment, we can see that there's almost the same particle size distribution to the whole sample with this particular shape. They show compressed microcapsule with some indentations due to the nozzle atomization pressure and the drying parameters. We use the same atomization parameters for the non-optimal, but we can see clearly that there's different shapes and particle size distribution through the sample. This is for the non-optimal treatment. Here's a zoom of the non-optimal treatment, and this is are the causes for the low encapsulation efficiency. We can see that some microcapsules are not completely formed and some broken capsules. That is why we saw a low encapsulation efficiency for the non-optimal microencapsulation. Also, we did confocal scanning laser microscopy. Here is the fluorescence spectrum. So for the components of the microcapsule, for free phenolics, here is the spectrum showing a green fluorescence, while the treatment show a blue fluorescence. Using this information, image A show the optimal treatment, and we can see the green fluorescence within the microcapsule. So in that manner, we can conclude that the bioactive compounds are within the microcapsules. This image to the right shows the non-optimal treatment, and we can see green fluorescence in these small particles indicating that are free phenolics. So due to the fact that there's some broken capsule, and also some of them are not encapsulated. After this, we can conclude that the microencapsulation of phenolic compounds from sugarcane bagasse was successful to protect them. Thank you so much. That will be all for me.
Thank you very much, Victor, for your presentation. Very nice presentation, very clear, easy to follow. And now we are open for questions from the audience. If you want to ask something to Victor, or if you want to write down your question in the chat, please go ahead.
Yes, good morning. I have a question for Victor.
Yes.
Victor, thank you for your presentation. I wanted to ask you, do you know the stability of your polyphenolic compounds after the spray drying?
After the microencapsulation?
Yes.
Yes, we did just the stability of the shell life by thermal treatment. So we measure the total phenolic content as a response variable. So it's stable during this time under this temperature stress.
Okay, thank you. And what can be the next step? So now you have encapsulated your polyphenolic compounds. What do you want to do with it?
Yes, it will be the digestibility test in vitro. That will be my next step.
Is it going to be with humans or with
No,
animals?
in vitro. First in vitro. Yes.
Okay. Thank you very much, Victor.
Thank you so much.
Did you measure the encapsulation efficiency?
Yes, I did. Sorry, let me. Yes, it's right here.
How do you compare the encapsulation efficiency of the spread dryer with the literature? What I mean by that, other encapsulation techniques?
Oh, it was very, very related. In some literature, I found that we can obtain from 60, 65 to 85. So my optimal treatment show encapsulation efficiency of 83. So there's not much literature related to sugarcane bagasse, but compared with other byproducts, some other plant extracts, it was very related to what I found in some literature.
Okay, thank you.
Victor, we have a question in the chat. That is, your sample from US has higher TPC and antioxidant activity compared to the sample from Mexico. However, antimicrobial activity is higher for samples from Mexico. How do you explain that?
Yes. Okay. Let me. Are you still able to see my presentation?
Yes, Victor.
Okay. So in this table we can see that the TPC from Mexico and from Texas are very related. There are no significant or major differences between Texas and Mexico, but Louisiana was more powerful compared with the other two. My explanation here is because I didn't have time to explain all of my research, is because here, while Louisiana showed more antioxidant activity and more TPC, was very low on antimicrobial activity, is because we found that after evaporating the solvent, the sample from Louisiana show like a candy-like aspect, like very sticky. So we did a measurement of sugars, and it showed a very high content of sugars compared with Mexico and Texas. That is why for bioassay tests, we use the lyophilized powder. So we took 2 milligrams of this powder. Then instead of taking phenolic compounds as a total, we took also some sugars because of this aspect. We tried to get rid of the sugars, but the molecular weight was very similar to the phenolic compounds. So we didn't take the risk of cleaning or taking the sugars without taking the risk of taking also the phenolic compounds out of the sample. But at this time, I cannot conclude that the Louisiana has no antimicrobial properties. I think it has, but it's because of the sugars still present in the sample.
Okay, thank you, Victor. Does anybody have a question for Victor? Okay. Well, in that case, we thanks to Victor for your presentation, and we're going to move forward to the next speaker. Thank you, Victor.
Thank you so
Great
much.
job.